that is now been used in almost every kind of research. So there are like several types of ANOVA. We will not go through each and everything for the today's lecture, but we will get through the basics and the things that before ANOVA we want to know. Also, we will go through a few little bit of R code and how we do it in the software. Okay. So, and in the towards the end of the session, we will also open the session for the queries. Okay. So let me proceed with the presentation. So this is a table of contents. Okay. And okay. So first of all, I want to discuss. Give me a second. So, what is basic purpose of ANOVA, or in a bigger sense, I will say, what is the basic purpose of this statistical significance testing, or all these? What is? Okay, okay. Sorry. Okay, sorry for the disruption. I think now everything is okay. Uh, we should proceed. So, what is the basic purpose of doing hypothesis testing, or um, in a simple sense, I can say, what is the purpose of applying statistics in first sense itself? Why, in a basic sense, we can't say that, okay, the mean is higher and the mean is lower, and directly can't we comprehend that this particular group is better, this particular group is bad, or particular treatment is better, particular variety is better? Why we cannot say just on the basis of simple mean? It is question with many people, that why it is necessary to conduct statistical analysis. So let me answer to begin the answer of this question. uh, we should understand that the difference in the group of two means in the difference, like here we can see, I have plotted, I have shown a figure in which the mean of the men group is quite higher and the mean of the women group is lower. Okay. So usually what happens when an experiment is conducted, we do take care of many things, like the setup is same, the in your experimental units are given similar kind of inputs, apart from the treatment that what you are going to compare. What is the reason behind that? Because we want to control the error that is being caused, that could have been caused by all these factors that are beyond the control of experiment. We want to minimize that. But even after you do minimize it by using a uniform setup, by using a all the things, uh, we cannot zero it out. Whatever you do, you can minimize, but you cannot zero it. So what we do in statistical analysis, basically, we account it in the form of variation. We account it in the form of error, because if you do not consider error, only then you will going to make the conclusion based on the direct means, which would not be just, because definitely you have controlled so many things, but you cannot keep a complete control on the inputs. We will come to the further examples. Uh, See what we firstly what we start with. Let's say if we have to compare two groups, one group is men, one group is women. Okay. So how do we proceed when it is two groups? Let's take a data. Okay. This is a library in R which is showing this. I would like to know if how many of the participants are acquainted with R software, if maybe in the chat box or whatsoever. No messages. So can you please say, like, out of the participants, how many of you have used R software before, or you have not even seen the interface or something like that? Anything? Okay. Reshma wants to say something. Okay. So you know. Okay. Okay, I think there is a little like miss. I don't know how much, but people are acquainted with the software. Feel like. Okay, so let me proceed with the uh, things here. How this the in this software there are majorly we go through means there are few things like libraries or packages. You can say same thing. In a package, there are so many functions that are being uh, stored in every package or a library, and then that library provides the facility to use that functions when we load that library. So library tidyverse, library rstatics, ggpabar, these are few libraries which are used for this particular example I am using here. There is a data of gender weight. Okay, gender weight data is there. It is available with the package Datarium. Okay, so let's look at the data. So this data has three columns. First column is ID column. Okay, ID column is your it is just nothing, just it is like a serial number. Okay, nothing more. It is just like a serial number, just it is naming of an individual, like first member of the female group, second member of the female group, third member of the female group, like that, like that. Okay, then second column is telling us which group to particular individual belongs. It belongs to female group or it belongs to male group. Like towards the end, you can see here it is not showing all the rows, only starting 18 rows, but in data you can see totally there are 40 rows are there. Okay, so some of them are male and some of them are female. I think 20 here. In the next table we have in the next column there is a weight. Okay, this is the weight of the uh, individuals. Okay, it is visible, right? Okay, so in the next table what we have done, next table here, what we have done is we have conducted the simple mean and standard deviation. Okay. So by just looking at the mean, however, you can say that the mean of female group is quite less than the mean of male group. Okay. Here it is 85, here it is 63, right? So, but when we have a two sample problem, these are two samples, so two groups, one is male, one is female. So the problem, when there is a problem of comparing two groups, two means, then we have to go for a basic t-test. Okay, so this is the formula of t-test. You can see on your screen, I think. I think there should be a bracket here. In this place there should be a bracket. Okay. So what we are testing in case of two groups, we are testing that the mean of first group is equals to the second group. And if this calculated value of this calculated value is higher than the table value. Table value is being calculated, is being taken from the T table. T table is nothing but the table from the distribution T. I won't go much into what is T distribution and all, but just you need to know that whenever we are calculating any statistic, whether T statistics, Z statistic, F statistics, then these are having their own distributions. T is having its own distribution, chi-square is having its own distribution, F is having its own distribution. So all of them are having their own distributions. Okay. So based on that distribution, it has some value on a particular degrees of freedom. So when we will be conducting our test will have some degrees of freedom based on the size of sample. Now in this case, you can see the size of sample is 20 and 20 in both. So total you have 38 degrees of freedom in this case. So this is the formula although, then we can do in R as well. Okay. Oh, the code is missing. Okay, okay. Okay, let me check once why the code went missing. Okay, let it be. Okay, okay. Just okay. I will share the code if it is not visible here. Let's see why it is not visible. But let's see. Let's have a look at the results. Okay. So what results you are getting over here is you are getting a t value. Okay. This is the t value here. T value is minus 20. Here degrees of freedom is 26. This is actually corrected degrees of freedom. Okay, corrected degrees of freedom is this is because advanced method is being used in R. So it is using corrected degrees of freedom instead of actual degrees of freedom. And here the p value is being written 2.2 e minus 16. Usually people get confused. What is this 2.2 e minus 16? E minus 16 means nothing but the number written in front, that is 2.2 multiplied by 10 raised to power minus 16. So when we will multiply 2.2 with 10 raised to power minus 16, it actually means that it is 0.00000, like that, 16 zeros, then 2. That is the meaning. So that means the p value is very, very, very small, and the computer could only calculate it up to the accuracy of 16 points. Okay. So based on the p-value also, see, I here in the last slide I have written that you can give the decision based on the comparison of calculated value and the table value. But the similar thing you can do by checking the p-value as well. What is the p-value method? Just you have to check if the p-value is less than 0.05, then you will say your test is significant. And here you can see the computer is telling you that what is your alternate hypothesis. Alternate hypothesis is that there is true difference in the means between group F and group M is not equal to 0. So that's what is your alternate hypothesis. So computer is stating computer is also stating the alternate hypothesis. The alternate hypothesis that you was using during the test, because in case of t-test there are three types of alternate hypothesis possible. So let's not go into that. Um, in this particular test, it was used that group F mean is not equal to group M mean. That true difference is not equal to 0. That was the um, hypothesis. So here your hypothesis got rejected because your p-value was less than 0.05. So when your p-value was less than 0.05, your test is significant. So when you say your test was significant, then you have to say that your alternate hypothesis, which was that mu1 is not equals to mu2, is considerable, and your null hypothesis, which is mu1 is equals to mu2, is not considerable. Okay. So this is basic t-test. Okay. When you have two group means. Okay. Then why, sir, you are telling all this? This is the lecture about ANOVA. This is, but you are telling about t-test. So why? Actually, what happens? The t-test is useful only up to the time when you have only two means to compare. When you have only two means to compare, then you have to use t-test. But whenever the number of means will exceed 2, that means if you have only two groups, male and female, then t-test is sufficient to use. But if you say you have more than two groups, like high income, medium income, low income, now there are three groups, or you want to compare three varieties, okay, or more than three varieties, maybe you want to compare 20 varieties, you want to compare 30 varieties. So in that case also, you have to go for ANOVA. You cannot stick to the t-test because the number of means has now exceeded. Okay. So that's how we proceed with the ANOVA. Okay. So let's now get into the ANOVA, straightforward. Okay. Now, what is the full form of ANOVA? Is, I think everybody knows ANOVA's full form is analysis of variance. And what is actually what is ANOVA doing and how it is achieving what we want to do? So basically, you can say there is total variance. Okay. Uh, there is a variability in data available. Actually, is there any way that I might get feedback from the participants? Like, I don't know if you people are getting, or I'm speaking from the one end, and maybe till now it is okay. Can okay, till now what I am up to, it is clear with you. Please say anything. You got it, you don't did not get. Students, hello. Participants.
Got it, sir.
Okay, okay, clear. Nice, nice. Okay, okay. So that's good, that's good. So now what is the thing? Now what is the thing? There is a total variation. Total variation means to say whatever the variability is available in your data, the total variability available in your data. Okay. So when the total variability you are considering, the total variability has all the components, that is the component due to um, your treatment. That means why you are conducting the test, you want to compare one treatment with another treatment, right? That is the purpose. You want to compare one group with another group, or you want to compare several groups. Okay, because of some variable. Say if you want to compare the yield of 10 varieties or the yield of 20 varieties, right? That means the fact you want to test, what you have as an objective to test is whether the difference in yield is coming due to change in variety. That's what is your objective you want to test. You want to know if the difference in mean is coming due to change in variety or anything else. Okay, because assume that you have set of 10 plots. Okay, in set of 10 plots you used two good plots. You know that these two plots are good plots and last two plots are bad plots. Okay, assume that you have 10 plots in total and two plots you already know, the first two, these are good plots, and the last two you already know that they are not good plots, they are bad plots. Okay, and while allocating the treatments to your plots, you allocated a treatment that you want to be better to the good plots, and the treatment that you do not want to be better you put into the bad plots. Okay, so now automatically there will be a difference in mean, but that difference in mean is not coming due to the variety. Okay, that difference in mean is coming only due to the difference in the soil fertility or the conditions prevailing in the plot, not due to the variety. But even though you do not want to do this, you want to be unbiased. Okay, this is the one scenario. This false scenario was a first scenario in which the person, the experimenter is biased. Okay, but in next scenario, I am presenting now new scenario. In this scenario, you say have same set of 10 plots with two good plots, two bad plots, and rest of the six are medium level plots. Okay, then in that kind of setup also, person wants to be unbiased, right? Person do not want a biased result. He wants to conclude the result in a unbiased manner. So can anyone amongst the participants say that how one can proceed? Anyone say, can something might have come to your mind? Say, anyone? Okay, nobody, huh? Okay, let's proceed. No problem. No problem. So here comes the thing that we say that ANOVA has the capability to partition the variation that is caused by your treatment and the variation that is caused by the factors that are beyond your control. Now, what are the things that are beyond your control? The things like the boundary plots are usually affected by the pests more, है ना? And there will be definitely a fertility gradient in your field. One side of the field will have a higher fertility, other side of the field will have a lower fertility, right? Then there will be a slope also. In the one side of the field, the water will be more stagnant. On the other side of the field, it rapidly it can be uh, drowned out. So there is the field conditions are not same throughout the plot, throughout the whole experimental setup, right? So those are the biotic and abiotic factors that are not under your control. Okay, right?
Yes, sir.
Is that okay?
Yes, sir.
Okay, okay. So now what are the assignable causes? The assignable causes are those which are under the experimenter's control. Like what? Experimenter's control, experimenter is only controlling the treatments. Experimenter is controlling the whatever the difference in the treatment will be there, whether it is the variety 1, variety 2, variety 3, whether it will be the different doses of the fertilizer, whether it will be the different doses of the pesticide. Okay. So one more thing I would like to know, guys, if you can say like all of us are from the agricultural background or like agriculture graduates or some other students are also there. Means I am sorry that I don't know this in prior. Uh, like or some other field also there. Agriculture students say yes.
Yes, sir. From agriculture background.
Okay. Yes. Okay. Now do those who are not agriculture say no. So I don't know. Okay. So I feel like most of us, most of our are from the agricultural graduates. So this otherwise I can change the type of example that I am quoting. That's why I asked, nothing else, because everyone should connect to the example. Okay. Then similarly, this is one assignable cause. The different fertilizer doses, the different this treatments, or all these things. These are the one type of assignable causes. Second thing, usually that comes in the assignable cause is blocks. Now, what is blocks? We will have a look in a further slide, the next slide maybe, that we will look into what is the block. Okay. So I think now we are clear what are the assignable causes and what are the non-assignable causes, and the ANOVA has the capability to partition the variation that is caused by assignable causes and the non-assignable causes. And based on this partition, we are able to learn that if the variation caused by the treatment is higher than the treat caused by the biotic abiotic factor, which usually falls into the error. This variation, this part of the variation, which is we are saying biotic and abiotic factors, this is going to fall into the part of the error. Okay, this will now be the part of error, and this will be the part of other sources of variation. Okay, so now let's see. This is the basic structure of ANOVA. Okay, basic structure of ANOVA shows you in this. Now this is the basic structure of basically one way ANOVA. Okay, we will discuss later what are the types of ANOVA, but in this slide we have put up a basic form of ANOVA. ANOVA is having, these are all calculated columns. Okay, so one thing is source of variation. This is the first column, sources of variation. What is the meaning of sources of variation? So as I said, there are so many sources that are causing variation in the experiment. We said assignable causes and non-assignable causes. So in assignable causes, we are considering treatment. Okay. So one source of variation becomes treatment, and whatever else other than treatment is there, that becomes the error in case of one-way ANOVA. In further, we will discuss what are the differences we will discuss. Okay. Now, second column is DF. DF is nothing but your degrees of freedom. Degrees of freedom, right? Then there is sum of squares. Sum of squares. I have not put the formulas and all for these things because I think otherwise that will become more of a theory class. But anything you have confusion in such things also, I'm open to answer. There is no issue regarding that. Okay. So sum of squares is nothing, but I think all of you are acquainted with variance. Variance, so everyone understands what is variance, I think. Okay, participants other than agriculture are also there, right? Okay, okay. Right. So, okay, what is variance? So already you are acquainted what is variance. So what is sum of squares? Sum of squares is nothing but the numerator part of the variance. Okay, you got this. Sum of squares is nothing but the numerator part of the variance. In a easy way, you can understand this thing. What is sum of squares? Sum of square is nothing but it is the numerator of the variance, right? Uh, and how you are getting this mean sum of squares? Mean sum of squares you will be getting by dividing sum of squares with degrees of freedom. You will get mean sum of squares. Then if you want mean sum of squares, you are dividing sum of square by degrees of freedom. Now your mean sum of squares is in a manner a representation of variance only. The mean sum of squares is representing basically variance that is caused by treatment, that your error mean sum of square, which will come here in this table, in this place, it will come. So this one will show you the variance that is caused by the error factors, and the treatment sum of squares, which will come here in this table, will tell you the variance that is being caused by the treatment, right? And how you will get f calculated. Now when you were comparing two means, that time the test that was used was t-test. But now when you are using more than, you want to compare more than two means, the test that is being used is f test. Okay, so now f test is nothing but the division of two variances. If you will check the formula for f, this is division of two variances. Okay, if the numerator variance is significantly higher than denominator variance, then we will say the test is significant and the variation caused by the numerator is significantly higher than the denominator. So in this scenario, what we want to check, you want to check if the variation caused by treatment is significantly higher than the variation caused by the error. So what we will do here in this place, you are getting the variance of treatment, and here in this place you are getting the variance by the error, and the f you will calculate based on the f you will calculate based on the division of variance of treatment divided by variance of the error. And if the variation caused by treatment is significantly higher, only then we will say the variation is actually caused by treatments. The difference in means is coming due to the treatments, and it is not coming due to the abiotic factors or biotic aphytic factors that are causing error in your experiment. So now let's discuss a little about this types of ANOVA. Okay, so what is the types of ANOVA? Here, when we are discussing the types of ANOVA, first thing, okay, here this should have came in the next row. Okay, no problem. This is together. Okay. So in one way ANOVA, you are considering only one source of variation, which is treatments. What is that? That is treatment. A simple example of a one-way ANOVA experiment is your CRD. CRD is completely randomized design. Completely randomized design. Why? because it is a single block design. Now, what is single block design? We will discuss in the next slide. Because for single block design, we need to understand what is the concept of block. So, for the understanding of concept of block, we need, I think, yes, okay. In the next slide, we will discuss. Then the two-way ANOVA. There is a second type of ANOVA is two-way ANOVA. The meaning of two-way is nothing but though instead of one source of variation in your ANOVA, there are two sources of variation. Two are, one is treatment and second is actually not replication, it should be blocks. Second is block. In case of two-way, it is the most common example that is in two-way ANOVA is RBD, a randomized block design. Randomized block design. That is the example of two-way ANOVA. In two-way ANOVA, there are two sources of variation. One is treatment, that is because the experimenter is now controlling two things. One is he is controlling the treatment, and second thing is controlling the fertility gradient in the field by making the different blocks. You'll understand. In three-way ANOVA, but this is the two-way ANOVA, and then we have a three-way ANOVA. So I would like to know anyone who is not acquainted with CRD RBD experiments. Means you people don't know what is CRD, what is RBD. From the means you have never read it. Anyone who has never seen or never read a CRD experiment on RBD experiment or whatever it is. Anyone who does not know? Okay, those who say who knows, say yes. Okay. No. Yes. No. Yes. Okay. 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 Oh, right, right, right. Right. Okay. Okay. So there are quite a population in our participants who do not know what is CRD, what is RBD, and what is LSD. Now, students, then I will like to say what is, in brief, I will let you know. Huh? Then we will discuss in detail, maybe in the next lecture or maybe in another session. What is in detail, we will discuss what is CRD, what is RBD, what is LSD. But for today, I will tell you a very basic difference in these experiments. Okay. So what is CRD? That is CRD full form is completely randomized design. So in completely randomized design, what happens? You have greater control over the error, over the inputs that you are being providing to the experimental units. Like how? Like you have a lab setup. In a lab setup, you are assume that you are doing an experiment in Petri plates. Okay, you are doing an experiment in Petri plates, and in Petri plates, you want to inoculate 5 or 10 strains of bacteria, and then you want to compare the growth in 7 days. Okay, you have 10 strains of bacteria and you want to compare their growth in 10 days or 7 days, in 7 days, how many colonies have came up. Okay. So what you will do, you will prepare a nutrient agar. Nutrient agar is a form in which we can grow this bacterial colony. Okay. Now assume that now you had 10 bacterial colonies. Okay. To 10 bacterial strains. So you will put, like, every strain you will put in 2 or 3 or 4 trays. Okay. Then you will prepare the nutrient agar for all these trays at once. Okay. So the input that you are giving now does not have that much of variation. Okay. Because all the petri plates are getting almost similar kind of input, similar kind of nutrient agar, equal amount of nutrient agar, and all of them will be kept on similar temperatures. Okay, that's what. Now, this is a setup in which there is no need to go for a second source of variation because many things are under control. So, you have sufficient CRD, and CRD as an experiment will be sufficient in such condition. That's why we say completely randomized design, because there is no blocking required in this scenario. But in case of two-way ANOVA, two-way ANOVA means to say RBD. RBD. Now, students of agriculture usually know that RBD is used in field condition. But why? Why not in lab condition? Why in field condition? Sir, field condition are not homogeneous. Now, what is the problem if they are not homogeneous? Let's see. Let's first go to the next slide, and then we will come back and discuss the rest of the types of ANOVA. See, look here. This is a figure from an experiment which was conducted in field. Okay. This is called a uniformity trial. Okay. What is uniformity trial? In a large field, what we will do, we will plant a single crop, or we do not give any treatment in this field, and we put up throughout the field, we put up the same crop, same variety, everything is same. Okay. Now, when everything is same, can you tell me, if we divide this field in these small plots like this, small plots like this, that this one dot comes in one plot. Okay. In such a manner, we divide this total field in a manner that every small dot will fall only in a single plot. Okay. Well, such a way you divide this whole field. If you divide this whole field in such small plots, okay, then tell me, there is no different trial, no different treatments being put. Then can you tell there will be the yield in every small plot will be equal or different? Tell me if it will be equal or different. Say. Yes, okay. Payal is saying different. Akhil is saying different. Okay. Shipra is also saying different. Okay. So, if Anyone, like Shipra, if you can unmute yourself and tell like why you think it will be different. Anyone can unmute and tell that why it is different.
Sir, fertility gradient and natural calamities and different types of treatment variation. If not human treatment, if not from the experimental side, it will come from other environmental effect will be there.
Right. Okay. Very nice. Very nice. So we are not putting any treatment here. Okay. We are not putting any treatment here, but what we are putting the same variety throughout the field. However, due to the difference in the soil type, the availability of nutrition in that particular small place, the availability of sunlight, the availability of water, and so many things. The maybe in some specific plot, the small insect might be there, which is impacting. That insect is not spreading in the whole field, but it is impacting in that small area only. Okay, so due to that, now you can see here uh, on the right hand side, there is a scale here. Here in the scale, you can see the greener it is, the higher is the yield, and the red it is, or more on the orangish side, lower is the yield, and the yellowish ones are the middle yield. Okay, so here you can see this red dot. This red dot is having, I think, very low yield. Okay, this one, this small plot which is covered by this orange dot is having, also having low yield. Here you can see, here there is a very green dot. This green dot is having a high yield. This green dot plot, the plot covered by this green dot, is having a high yield. Okay, but see now this yellow. These all yellows are almost nearby. So these are almost having similar yield. These yellow. This is also these having similar yield. But this is orange. These are having lower yield, but all the orange are similar to each other. All the reds are similar to each other. All the yellows are similar to each other. All the greens are similar to each other. So what you learned, there is a variation in the field. Okay, that we know. That is the main thing. After conducting a uniformity trial, you already know that there is such variation existing in the field. Okay. Now, when you know that there is a variation existing in the field, you must include this into the information in the form of information given in ANOVA. Okay. Now, how we will feed this information to the ANOVA? We will consider it as a source of variation. The fertility, which is changing, or the not the fertility, you can say the if we group these plots, the yellow ones in first group, the green ones in second group, the orange ones in the third group, the red ones in the fourth group, like this. Okay, or the lemon color ones in the fifth group. So what we made five groups. Now these five groups, within each group they have similar characteristics, but in between there is a difference. Okay, the green ones are having similar yield, the yellow ones having similar yield, red ones having similar yield, but the yield of red ones is different from the yield of green ones. The yield of green ones is different from that of yellow ones. So what we say, we construct a block. What we construct? We construct a block which is consisted of similar characteristics plots. All those plots having similar characteristic, we put in one block. So the green ones fall in one block, yellow ones in one block, red ones in another block, orange ones in another block, lemon colored in another block. Okay. And when we allocate our treatments to the plots, we also note down which category of block this particular plot belongs to. Okay. So anything, if it got little more complex, please you can ask. Okay. Anything you got if you anyone if you got little uh, you did not get me, you can ask a question. Anything. I will otherwise I will proceed. Guys, is this clear? What I told? What is the meaning of block? You got it? Others, guys? Oh, very nice. Very nice. Very nice. Okay. 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 Good. 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 Okay. So definitely you understood that the green dots fall in single block, the red ones fall in second block, like that. Okay. Then this block will also become the source of variation in two-way ANOVA. Here, this block will also become a source of variation, because even after you control all the inputs that you will supply, this block will also cause somewhat of variation in your ANOVA. In total variation, some of the variation will be caused by this factor which you call block. Okay. So that is when we, that's what we call two-way ANOVA in case of RBD. That is two sources of variation. One is your treatment that you want to compare, and second is your block. Okay. Now we will there is three-way ANOVA. This is actually less used. This is LSD, Latin square design. Latin square design is relatively used less because it consumes a very much resources. Where if they, because the number of treatments, the number of replication, the number of times you need to repeat a treatment is equals to the number of treatments. So if you have 10 treatments, then you have to repeat each treatment at least 10 times to conduct this three-way ANOVA. Okay. So in total, you would have to run 100 experimental units if you want to compare 10 treatments. So it is a very resource consuming design. That's why it is usually not very much used in research. But the thing is, it is useful when, apart from block, also there is some other source of variation existing. then this is useful. We say when there is a two-way field gradient. Okay, we don't go much into what is two-way field gradient today. We will proceed with the lecture, otherwise the thing concept will remain. Okay, so the next type of ANOVA is factorial. Factorial design is to say what is the when you have more than two type of variable you want to compare at a time, you can also consider their interaction. What is the meaning of interaction? Assume that there is a experiment in which there are five group of people. Okay, five group of humans. There is a person, there is an experimenter who wants to test if there is a difference in one type of sleep cycle, second type of sleep cycle, and third type of sleep cycle on the productivity of a human. Okay, person want to test, experimenter has the objective that he want to know the people who sleep 6 hours a day are more productive, who sleeps 8 hours a day are more productive, or those who sleeps abruptly are more productive in their day. Okay, side by side he also wants to know if the dietary habits, diet 1, diet 2, and diet 3, how diet is affecting the behavior of the human in an experiment. Okay, now what he could have done, he could have gone for a one-way experiment and used sleep cycles as treatments and used it in three groups. Also, again, he could have used the second treatment. He want to check the diet 1, diet 2, diet 3, and he would have given these three diets to three different groups. And in both the experiments, he could have concluded, okay, sleep cycle of type 1 is the best sleep cycle. And he could have concluded that diet 1 of the all is the best type of diet that is suitable for all the humans, that is most productive for the day in case of this group. But it is not necessary that diet and sleep cycle are not independent. They have some relation with each other. Means I am not saying that they have relation. Means there might be, there might not be, but it needed to be tested. That is the thing. One needs to know if there is any type of dependency between these two, the dietary habit and the sleep cycle. Then he has to set up an experiment which is called a factorial design, in which he will use two-factor design, in which one factor will be sleep cycle and second factor will be the dietary habit. And then he will need 9 groups while making the combinations diet 1 and sleep cycle 1, diet 1, sleep cycle 2, diet 1, sleep cycle 3, diet 2, sleep cycle 1, diet 2, sleep cycle 2, diet 2, sleep cycle 3, and diet 3, sleep cycle 1, diet 3, sleep cycle 2, diet 3, sleep cycle 3. Like that, 9 groups will be there to which he will allocate the experiment to, and in that experiment then he will be able to conclude if there is any relation between these two factors. So when he is going to study the effects individually, that is the effect of diet and the effect of sleep cycle individually he is studying, that will be called main effect. And when they will be studied together, that if there is dependence or not, that will be called interaction effect. Okay, so then it will come under the ANOVA of factorial design, in which there is a interaction effect that is being considered. Then there is a repeated measure ANOVA, that is in case of dependent variable, dependent observations. I think let's not go much deep into that. That is the part of populations when you have to take repeated observations on the data. Like if you have an orchard, okay, in orchard you in the first year applied the first type of treatment. In the second year, for the same tree, you are applying the second type of treatment. In the third year, for the same tree, you are applying the third type of treatment. That means the subject to which you are applying the experiment is remaining same. It is not getting changed. In that kind of cases, you need repeated measures. Otherwise, the name makes it clear that you are making the repeated measurements on the same subject. That type of ANOVA is called repeated measures ANOVA. For today's lecture, we will keep it up to the one way in example. Then we will proceed in the further sessions. We will proceed to the two-way and the factorial designs. Repeated, if we have some time for the repeated measure, we will keep that one as well. Okay, so let's now, I think this one we have discussed already. So let's now, okay, there is one more important part I forgot to discuss. This is called assumptions of ANOVA. What are the assumptions of ANOVA? The three assumptions of ANOVA. Okay, one is called normality, second is called additivity, and third is called homogeneity. These seems like very typical terms might be to all of you, but they are not as typical as the names seem to be. Okay. So the normality of population means what? The normality of population means that your treatment one, when you took the reading, you took the reading on 10 units, you took the reading on 5 units, you took the reading on 6 units, maybe any number of units you took the reading. What is the assumption saying? That when you were taking the observation of 5 or 3 or how n number of units for treatment 1, the observation of the observations was the sample out of a normally distributed population. That is the assumption. Similarly, for the treatment 2, also you were taking a sample out of normal distribution with mean mu. For first population, it was the mean mu1. With the second population, that is the second treatment population, is the mean mu2, and similarly mean mu3, mu4 for all the treatments. If you have 10 treatments, then you are taking the sample out of normal distribution with mean mu1, with mean mu2, with mean mu3, like this, like this. So every time you was taking a population, you were taking observation, it is assumed that these observations were observed, that were a sample out of a normal distribution, not any other distribution. That is the assumption of normality. Okay, is it clear? Normality assumption. Normality assumption is clear, or I should say it once again. Tell me, should I say it once again? Okay, once again. All right, right. So firstly, I would like to know if you know what is normal distribution. What is normal distribution, guys? You are familiar or not? Not clear. Not exactly known. Okay. Anyone else? Normal distribution. Okay, I will tell. I will say what is normal distribution. So what does normal distribution in a very simple sense, I will say. I think you might have seen the graph of normal distribution, that bell-shaped graph. But what that bell-shaped graph is telling, that I will tell you today. That bell-shaped graph is saying that, it is telling you that the most, that is 60% of the data of the total population, not exactly 60, actually it is 65 point few digits, 65% roughly you can say. And 65% of data lies within the range of mean plus standard deviation and mean minus standard deviation. That is, if mean was 5, mu was 5, standard deviation was 2, then 5 plus 2 is 7 and 5 minus 2 is 3. That means 3, from 3 to 7. Assume that if it is a weight of, say it is the weight of seed in grams, then what the normal distribution is claiming, that out of all the seeds, 65%, 67% of seeds will remain, will have the weight in between 3 gram to 7 grams. And 95% of the population, 90, this is 67% of the population, okay, then 95% of population will lie in between mean minus 2 times standard deviation and mean plus 2 times of standard deviation. Okay, so what was the mean? Mean was 5. What was the standard deviation? Standard deviation was 2. So 2 times standard deviation means 4. So 5 minus 4 is 1. And 5 plus 4 is 9. So your 95% of seeds will have weight in between 1 gram to 9 grams. Okay. Similarly, 99% of the population will lie between mean minus 3 times of standard deviation and mean plus 3 times of standard deviation. That is a basic definition, very basic way to understand what we mean from a normally distributed data. Then there are several properties that mean, median, mode are same, and it is symmetrical on the both ends, and this and all is technical then. Okay, and so many other properties. But this is the basic thing, that what we mean when we say it is a normal population. Now, normal population is clear. Guys, okay, very nice, very nice. So now what we are assuming in case of ANOVA, that is, we are assuming that when you are taking a 5 replication, we are taking 5 repeated readings for treatment 1, then all 5 will come from a normal distribution with mean equal to the mean of treatment 1 and standard deviation of treatment 1. Similarly, for second treatment, it is coming from second normal distribution with mean equal to the treatment mean of second treatment. Then third treatment also, fourth treatment, the number of, and up to the all number of treatments, it is in this manner. Okay, better now? It is clear? It is better now? You got it? Normality? Okay. Okay. Now, okay, then we will proceed for the second assumption, that is additivity of the effects. This should be additivity actually, additivity of the effects. Additivity of the effects, you want to say that when you are considering this, here you said no, there sources of variation, there is treatment and error, then you are telling total, right? You are telling treatment, then you are telling error, then you are taking total. Why you are not multiplying the treatment with error? That could also be possible, no? Why you are not doing the square of treatment and adding to the error? That could also have been done. The formula could have been anything. But what we are doing, we are taking the total of the sum of squares of the treatment and sum of squares of the total. Okay. We are taking in total. Also, this is not only for this uh, treatment and error. Also, we want to say that treatment effects are additive. Okay, that is when you are going from treatment level 1 to treatment level 2, that is, say if you have different doses of some poison, say some, you are taking 10 ppm of first poison, 10 ppm of chemical 1, then you are taking 20 ppm of chemical 1, and then you are taking 30 ppm of chemical 1, then the it is causing death in a linear manner. Okay, so treatment effects are additive. The treatment effects are additive. Okay, and the third assumption is homogeneity of the variances. Homogeneity word you are familiar already. Homogeneity means it is similar. In English, if you translate the word homogeneity, it means similarity. Okay. So homogeneity of variances is just like saying that although the mean of different normal populations was different, the variance, if not equal, it is similar. You have 20 treatments. All the treatment might have different mean. That is not issue. But the variance, normal distribution has two parameters. One is mu, second is sigma square. Mu means mean and sigma square means variance. So when you are telling that all the populations, all the treatments are coming from different normal populations, normally distributed populations, then these normally distribution populations have different mu, different mean, and different standard deviation. The third assumption, the homogeneity of variances, say that the means might be different, but the variances of these population is not very divergent. It is very much similar. Okay, it is not claiming that it is equal. It is not saying that every variance should be 2, 2, 2, 2, 2. No, it might be 2, 2.5, 1.5, something like nearby. They should be similar, homogeneous in nature. Okay, this is called homogeneity of the variances. Then what is the last word I have written in slide is transformations. Transformations means what? If these assumptions are not met in real life data, what then you have to do? If the assumptions of this data are not met, then what we can do in that particular scenario? In that scenario, we will transform our complete data. What is the meaning of transform? Means we will apply some formula, some say we will take the log of all data, we will take square root of all data. Okay, such things we do, so that if particular assumption is not met, then we will, after taking such a transformation, then we can recheck if the assumption is met or not. For checking if the assumption is met or not, there are several tests. Uh, we will discuss, we are not discussing those tests for today. Otherwise, it will get very lengthy. We will see if we have time some other day. Normality, additivity, and homogeneity, all this we can test, and if any test is failed, then we will go for transformation. Then using transformation, again we can test if assumption is met or not. If assumption is met, then okay. If assumption is not met, also there is solution, then we can go for non-parametric tests. But that is not the topic today. So for us, let's assume that the assumptions has been met. Okay. So example, let's move for an example. ज़्यादा lengthy हो गया क्या? It is getting, anybody now, you people getting bored? Huh? Or you are with me? You are still with me? Or slept in between? No, it's going. Okay, okay. More. Rest. Okay, okay, okay. Then let's, I think I have two warriors. Still, they are listening. Okay, okay. We have more. Nice, nice, nice. Okay, okay. Very good, very good. Okay. Then we will keep on. Let's discuss one example. Okay, this is a data set. Now what it is saying, 12 homeowners are selected randomly to participate in an experiment with plant nursery. Each homeowner is asked to select four fairly identical areas in his yard to plant four types of grasses in each area. Now, what is happening in this? Now, uh, there are 48 observations. Okay, 48 how? There are 12 homeowners. Okay, so everyone is having four plots. Okay, every like everyone having four different plots, in which for every first plot is putting first type of grass, second it is putting second type of grass, and then it is putting third type of grass and fourth type of grass every person is putting in his nursery. Okay, so these 12 people, each person is putting four type of grasses in his area. So 12 person, every person putting four. So in total it became in total it became 4 48. 12 into 4, 48. Okay, so 48 observations you have, and then they recorded the data. Okay, in here which grass is planted, that will come in the treatment. Okay, the farmer number is coming in the column 1, that is judge, and the evaluation done, after that he is giving the score. Okay, that is coming in the last column. Okay, now let's check the oh, here also the code is missing. Okay, I'll share the code. Okay, I will share the code as well. I will share the another screen in which the code is being visible. Okay, in this I don't know why the code is not visible in this particular, but code is there. So in this, what is happening, the ANOVA is being conducted. Here we got degrees of freedom 3, residual degrees. This residual is telling you nothing but the error. Okay, this residual is a error. Then after degrees of freedom, again you are getting sum of squares due to the treatment. Here you got mean sum of square. This mean sum of square is you are getting by dividing the sum of square with the degrees of freedom. Okay, and here also the error mean sum of squares you are getting by the dividing sum of square of error with the degrees of freedom of the error. And finally what you are doing, I told you, you are doing the F test. F test is done by dividing the variance. This is now the variance, dividing two variances. This is the variance, this is the variance. So the variance of treatment divided by the variance of the error. Now you got this value. So this value is then compared to the table value. And what is the p value? P value is the probability of occurring of this particular f value given this experimental data, given this experimental setup. So if the p value is less than 0.05, if it is less than 0.05, it is said that it is significant. Okay, so that means our null hypothesis says that there is no significant difference between 4 type of grasses, but your alternate hypothesis, that is the second hypothesis, is telling, which is if your test is significant, that means if the test is significant, it is telling us that means are different. At least one of the mean is not equal to the other means. There exists at least one grass which is not equal to the other grasses. Okay, so let's now move forward here. Okay, um, this is now what we are doing. This is called LSD test. LSD is called least significant difference test. Okay, least significance difference test you conducted for the model. Here it is showing the code. Why it is not showing code here? Okay, let's see why it is not showing code. Don't worry. Okay, here then we are getting three major parameters. One is this. One is CV. This is CV. CV is quite high. Okay, CV is quite high means there is need of blocking. If your CV is high, CV is called coefficient of variation. Coefficient of variation is calculated based on the mean and the standard deviation. If coefficient of variation is high in any experiment, that means blocking is not done properly. There is a need to conduct proper blocking. Okay. Then there is LSD. This is LSD. That is CD value. Okay. CD value. CD is critical difference. Now, this is just like a standard. Critical difference is a standard, that is, you will compare two treatments based on the critical difference. LSD value, that is CD value. LSD value and CD value are same thing. Okay, least significant difference. What is the meaning? Least significant difference. 3 words. That means the minimum difference that must be there between the means of two treatments, so that we may call them significantly different. That is your CD or LSD value. So now let's see 0.82. Okay, so 0.82 is, now let's say 3.16 is the treatment 1, mean of the treatment 1, and 1.95 is the mean of treatment 2. The difference between mean of treatment 1 and treatment 2 is near about 1.2. Okay, the difference between the mean of treatment 1 and treatment 2 is near about 1.2. So what is the CD value? CD value is 0.82. And what is the difference between the mean of treatment 1 and treatment 2? It is near about 1.2. Okay, this is calculation I am making on my own. Okay, so this is now 1.2 near about. So this 1.2 is greater than your least significant difference. It is greater than the minimum difference that is required for two treatments to be significant. So when it is more than the minimum difference, then we say treatment 1 and treatment 2 are significantly different. But you see here T1 and T4. Okay, T1 and T4. The difference between the mean of T1 and T4 is nearly 0.23, 0.3 something, 0.29. The difference between the mean of treatment 1 and treatment 4, that is the grass number 4 and the grass number 1 is 0.29, but the least significant difference, that is critical difference CD, is 0.82. 0.82 is higher than the value 0.3, 0.29. 0.29 is smaller, 0.82 is larger value. So the difference between the mean of T1 and T4 is smaller and LSD value is greater. So you can say that the treatments T1 and T4 are not significantly different. That means the grasses T1 and T4 are almost similar in their characteristics, and the difference in their mean is coming only due to the abiotic factors. Okay. Is this clear? Okay. More? Right, right. So what we do here, there is the presentation. This is the way to present it. This is the way to present. Okay. This is how we present it. Okay. We are presenting T1, T4, T3, T2. This is the mean. Okay. And here one letter is being written. So you see T1 and T4 both has letter A. Okay. The letter, if two treatments share a letter, that means that letter is present for T1 also and T4 also. Then it is said that these are not significantly different as we have tested before. But see, is there any common letter between T1 and T2? In T1 it is only A and in T2 it is only C. So T1 and T2 are significantly different because they do not share any common letter. Similarly, can you tell T1 and T3, are they significantly different or not? Can you tell T1 and T3, are they significantly different or not?
Different.
Yes, they are different. Why? Because they do not share any common character. Because they do not share any common character. Also, the difference between their mean is greater than the lowest significance, least significant difference. Is this clear? So if it is clear, then we will move towards the end of the session. Okay. We will make a plot for this. Okay. Let me show you the plot. So this is the plot. Okay. So this is very basic plot actually. We can improve this plot quite so much. This is I did it just last moment. So this is this dot in between. It is showing the mean of the experiment, mean of the grass 1. This one is showing the mean of grass 4, mean of grass 3, mean of grass 2, and these are the uh, error bars, you can say, or the minimum or maximum values. This is the minimum value, the maximum value of the T1, and this is the minimum value of T4, the maximum value of T4. This is the minimum value of T3, the maximum value of T3, minimum value of T2, and maximum value of T2. Okay. So, but this is I also said that I will show you the code for this one. So let me see. Wait, I need to refresh my page. Give me a few minutes. And, let me share it again. So this is the code for t-test that we conducted in starting. Okay, this is the code for t-test, and this is the code for ANOVA. Okay, this in some further session we will discuss that what will come on this side. Okay, and what will come on this side, what is this, everything we will discuss. Okay, in further sessions we will discuss this and the graphical portion. This is the code to conduct LSD for same ANOVA, and this is the code for plotting this particular type of graph. So with this, I will open this session for the questions, for any queries. Anything, any queries you want to ask me, I am here to answer. I am stopping the screen share if you want. If anyone, if you want these codes and all, I will share, no problem. Okay.
Thank you, Dr. Saurav Singla, for this very interesting as well as insightful presentation. I think our participants will have a great benefit out of this presentation, and also hoping for your next session. And all the participants are requested that if you have any question, you can put it in that chat box, as well as you can also send those messages in our WhatsApp group, so that we can convey these things to Saurav sir for the next class, so that he can address it. Thank you, everyone.
Yes, yes, yes. Thank you.